basically for a modulated drug delivery, where you can have different drugs loaded in individual compartments, and you can actually tune the release based on the need and the real feedback from the patient, right? So now in terms of market opportunities and significance, because we all know 3D printing is really booming, especially for global medicines market. There's huge opportunity because the global medicine market surpassed about $1 trillion in revenue in 2014, and it is expected to be doubled in the next couple of years. These are some statistics that shows that 3D printing is gaining momentum across industries, be it in surgery or artificial model development for operations, or in the consumer goods products, military crafts, etc. So 3D printing is really getting its access to all industry sectors. The market size in medical industry, 3D printing market is really becoming large. So it is expected to be about 32.78 billion by the next 1 or 2 years. And interestingly, out of that, about 60% is for medical, like where we, this is an area where we have most interest, right? So about $5.24 billion. And most interestingly for us in the US, we have about 91% captured alone from our region over here when you talk about medical application or medical sector for the 3D printing. So overall, in summary, 3D printing has a huge opportunity for market and significance. So now let's take you to some quick case studies before I wrap up my presentation, and then I can open up the floor for some questions and collaboration opportunities or whatever. So there are two segments in my case studies. The first segment will be about personalized drug delivery concept by 3D printing. And the second one is about how to commercialize 3D printing based technologies or 3D printed products based on based on the opportunities for commercial exploitations. The first case study is about personalized 3D printed tablets, oral dosage forms. So this has already been published. So I'm not going too detailed in science. Rather, I'm more interested in showing the overview of the applications of 3D printing for personalized dose. So in this one, we used a poorly water-soluble drug as a model drug. So here we wanted to promote, or we wanted to prove that 3D printing can be used for tuning or personalizing the release profiles based on the need. And in this case, we only controlled the structure of 3D printed products. And by controlling the structure of printed tablets, we could actually control the release or personalize the release of the drug. So this is the overall schematic diagram of the process that we used. So we combined hot melt extrusion with an FDM printing based concept. We used hot melt extrusion to produce thermoplastic polymeric filaments containing drug in a solid dispersion form. And then we used that feedstock materials through the field deposition modeling based printing to produce the tablets of interest. So our target was to produce various tablets with different kind of porosities. So there is one term which is widely known to those who are familiar with FDM based 3D printing, is that it's called field density. So field density is a term that actually denotes, or that actually specifies how much materials are being deposited. So as can be seen, field density means like the higher the value, the denser the tablet. So in this photograph over here, the left column is basically with 20% field density. That means we are only filling 20% of the whole surface, whereas the second one is 40%. So that means we are actually filling 40% of the whole surface. The third one is 60%, and the last one is 80%. So as you can see, the higher the field density value, the denser the tablets, right? And then after producing the filaments, we did some comparative mechanochemical characterization, and what we found that our produced filaments, in terms of their gradient value, flexibility, and hardness, they actually were between the upper limit and the lower limit. So used actually two different reference materials taken from commercial, taken from the market basically, and we used them as soft and hard reference. And what we found that our produced filaments, they actually all fell between these two range, so which seem to be printable, right? And now, by only tuning the field density, or by taking the field density as an effective tool to control the release or modulate the release of the drug. So we kept basically the drug content the same, like 20% weight by weight, and we used the same polymer, 3 different grades. We used hydroxypropyl methyl cellulose, HG, MG, and LG grade. So like higher, medium, and lower in terms of the molecular weight. And what we found that by only controlling the field density, we could easily control the release pattern or release profile of that poorly water-soluble drug in basic pH at 6.8. So as can be seen from the photograph over here, the top panel over here, that the higher the field density, the lower the release, because you are actually creating more dense tablets. Most importantly, when we actually change the grade of the polymer, what we found that we could also control the release pattern by changing the molecular weight of the main formulation composition, the main polymer being used in the formulation composition. So this was really, really good example case study of using 3D printing where field density could be used for controlling the release profiles. We also conducted advanced analysis during the dissolution studies to see whether there was an evidence of recrystallization with drug content 20%, and we found that there was no evidence of drug recrystallization when the samples were withdrawn during the dissolution studies, and that confirmed that our adopted technology actually produced the drug in fully in its amorphous form. Right, so this was a good example, but I'm not going in detail because we have already published this paper. If you're interested to read, you're more than welcome to obtain this from this link. So the second case study is about producing 3D printed thin films for personalized application. So the reason why we conducted this study was mostly to show the viability of 3D printing for producing thin films, right? So we compared with some other traditionally adopted technologies like melt casting, hot mill extrusion, and then, of course, we used our 3D printing just to look at the structure and also the properties and performance. So as can be seen from this bottom panel over here, right-hand side, when we did the basically mapping of the deposited or loaded drugs, what we found that for the melt casting, there were some regions where enrichment of the drug was evident. So that means some drugs were deposited into an irregular manner. So in some area, drugs were more, whereas as opposed to that scenario, there were some other areas where drugs were not that much, drug was not present in that quantity. So which was mainly due to the absence of mixing process in the melt cast. Similarly, hot mill extrusion based film actually showed better homogeneity in terms of the drug distribution. But when we compared with our 3D printing tablet, 3D printing dosage form, we found that drug distribution was much better in our 3D printed film. So when we went for doing this advanced physicochemical characterization, especially the advanced surface analysis of the drug distribution, what we found that in our 3D printed films, drugs were present in amorphous form, and the distribution of the amorphous drug onto the surface of interest was more homogeneous compared to the other films printed by other technologies, right? And that was mainly due to the high shear mixing of the extrusion and then the 3D printing process. And most importantly, as can be seen from this photograph, drugs were present in its amorphous form. And as a result of that amorphicity, what we found that when we did the dissolution studies in both simulated saliva as well as the phosphate buffer, we found that the drug was released faster from our 3D printed film compared to the other traditionally manufactured films. So that actually shows a good example of how 3D printing can be used for enhancing the dissolution rate of the drug by forming amorphous solid dispersion. We also published this paper recently and it is available as well. So this is another good example that we recently published. So actually about a week ago, we published this paper, and for the first time, we wanted to use 3D printing concept for the development of a container system. So container system is good for kind of cargoing or delivering highly sensitive drugs, so like which are not stable in stomach pH or acidic pH. Like this concept can be used for successfully cargoing or shipping drug to the intestine. So we actually designed this kind of container platforms where it is quite intact. There was no holes or aperture. We only included one aperture of interest over here so that drug can be released through this aperture once it goes into intestine. So we actually loaded multiple drug containing SNAPs formulation. Here we use two different drugs, like one is curcumin, which is poorly water soluble drug, and the second one was lansoprazole, which is a PPI that is highly unstable in acidic condition. And we did some physicochemical characterization of the blank and drug loaded SNAPs, and what we found, the antioxidant level was dependent on the formulation composition, and the effective, like kind of like 50% effectiveness was achieved at about 330 to 393 microgram per milliliter concentration for both SNAPs formulation. So the idea was basically to prove a concept that a 3D printed container system can be used to load ultra sensitive or highly unstable drug molecules. It can be used for biologics in the future. If you wanted to deliver proteins or peptides orally, that can be an ideal solution. And of course, we did the SEM imaging as well as micro CT imaging to see whether there was any leakage or anything. And we found that our developed chamber system or container system was quite content in terms of the printing, I think the composition or the morphology of the container system that we have developed. And this is basically to show the release pattern of the drug. What we found that by simply controlling the wall thickness and the container composition, we could easily delay or control the release. So that may be an ideal paradigm for biologics formulation or unstable formulations in the future. We have just published this paper last week. So this is another example. So that is just to show how magnetically triggerable anti-cancer drug delivery system can be developed by using 3D printing strategy. So here we developed macroporous sponge, PDMS-based sponge loaded with magnetically triggerable particles, and then we loaded anti-cancer drug into the sponge, and we used actually 5-fluorouracil as the model anti-cancer drug. And the idea was basically to use magnetic field as a triggering factor for kind of like squeezing that macroporous sponge to release the drug on demand. So that can be an ideal solution for long-term treatment or long-term delivery of chemotherapeutic candidates in the future. So where a drug-loaded sponge can be implanted, and then you could basically release the drug by switching on or off of the gate from the macroporous sponge. So these are some characterization data in terms of their compressibility, mechanochemical information, as well as solid state analysis. We also published a couple of papers by highlighting this magnetically triggerable anti-cancer drug delivery systems. So we have done it. If you wanted to get an access to these papers, just let me know. We can certainly send them over. So our main focus was to look at like how precisely could you control the release from the from this chamber or from this magnetically triggerable 3D printed drug delivery system, and what can be seen from this drug release study over here. By switching on or off, you could easily modulate or control the release of the drug from the system. When we did the in vitro cell culture study on different cancerous cell lines, what we found that when the formulation was truly optimized, we could actually inhibit the cancerous cell growth significantly. So this was a really good proof of concept study to show that 3D printing can be successfully used for magnetically triggerable drug delivery systems. Right. So now I would actually take about 5 to 10 minutes more to go over some platform technologies and see how 3D printing can be used for producing platform technologies with true potential for commercialization, right? So up to now, we have mostly talked about personalization and the real opportunities of using 3D printing for personalized drug delivery. But now in this segment, I'll be focusing more on the commercial possibilities, or using 3D printing for commercially viable platform technology development, right? So up to now, you might have already heard that 3D printing is relatively slow process. It takes about 5 to 10 minutes for printing one tablet. And the question is, if you wanted to produce millions or billions of doses, how long would that take? So would that really be viable? That was one of the major questions lately. So in order to overcome some of those problems, in order to answer some of those questions, we thought of exploring other viable options for commercial exploitation. So one example is the selective laser sintering based printing option. Here in this first case study, we use ritonavir, which is an antiviral drug for HIV, and we use 10% and 20% loadings. And ritonavir is a highly unstable and heat sensitive drug. It has very poor solubility. So we used actually our selective laser sintering based concept for the single step production of amorphous tablet without jeopardizing or compromising the stability of that drug. So these are some of the key process parameters that can be adjusted and tuned to produce successful tablets. So I'm not going in detail, but this is an equation that actually shows how much energy is being produced by the laser that actually sinters the particles to form 3D printed constructs of loosely bound particles. We have published this paper. We have also filed a patent applications on that. And our published paper was recently highlighted in a special topic cluster from Journal of Pharmaceutical Sciences. So this table actually shows various formulation compositions we trial and error. We developed in a trial and error method. And this video actually shows very quickly like how the printing is actually works. As here, as you can see, that laser beam is actually being deposited on selectively onto a selective area of powder, layered by this blade over here, and then it is actually fusing the particles through the layer by layer fusion and through the energy being actually produced by the laser over here, and then once the first layer is sintered completely, then another successive layer will be deposited onto the previous layer so that the process can continue repeatedly, right, until we get the final tablet of interest. That's how you see the another layer is now being deposited onto the previous one, and this process will keep going until you finally develop the tablet. So this is really, really a promising technology for single-step manufacturing of 3D printed medicine. So these are physicochemical characterization, shows that after the single-step process optimization, the highly crystalline ritonavir converted to its amorphous form, and there was no evidence of crystallinity. We also did advanced characterization for the amorphous nature and intermolecular interactions analysis by 1D and 2D NMR, solid-state NMR analysis. And what we found that our drug was present in amorphous form after the SLS process, and there was a higher degree of misstability in our 3D printed dosage forms that was reflected by the shorter relaxation time. So, and most importantly, we found that our 3D printed tablet actually showed 21-fold increase in dissolution of the poorly water soluble drug compared to the physical mixture or the bulk drug. So it was really, really good summary of our technology. And then we wanted to move further. So in the previous case study, we used SLS as a single step process in a segmented or batch manufacturing. So we wanted to actually combine that or establish that into a truly continuous digital manufacturing platform. And that's why we came up with the dose plus technology, where traditional twin screw extruder or twin screw extrusion process was combined with a custom made SLS printer. And then you could actually print tablets in a single step digitally interconnected manufacturing process. And here the speed and the scale can be easily large. And it really depends on how big you want to go up to and what would be the scale. So this is truly viable for commercial exploitation. We published this paper. We also filed patent applications on this as well. And we are currently discussing with a couple of pharmaceutical companies for its commercial exploitation. This is just a short video to show like once the tablets are printed, see how fast it disintegrates. Because the faster disintegration is due to the porosity, the tablets that we produce through the laser sintering process. And these tablets, they actually disintegrate within about 3 seconds without any agitations. The moment you introduce water, it disintegrates. So the reason is that, okay, the tablets we produce, they're highly porous, and the porosity ranges, or the mean value of porosity is about or the void structure is about about 142 micrometers. And as a result of that and the single step manufacturing of amorphous solid dispersion, we found that our tablet showed much better release compared to the counterparts or comparative formulation. So that was really good indication of proving the concept that our dose plus can really be used for enhancing the physicochemical properties of the poorly challenging molecules. So we continued doing more investigation. We looked at the effect of laser speed, drug particle size, and by looking at all the examples, what we found that our dose plus was really a viable option to not only produce amorphous solid dispersion, but also maintain its amorphosity even during supersaturated condition-based dissolution studies. And from this study over here, as can be seen, that when we withdrew the sample, we drew the sample from the dissolution vessel in a highly supersaturated condition. There was no evidence of recrystallization. So that was really great uh, output based on the application of our technology there, right? So we also recently used this for photosensitive drug as well, and this was recently highlighted as the cover photo of molecular pharmaceutics. So that was really another good indication of our technology. And recently we published this paper in Molecular Pharmaceutics. Actually, we published it just about a day ago. And here we collaborated with the University of Cambridge to look at the trace crystallinity of the drug. And what we found, even though the drug content was high, there is no evidence of trace crystallinity from our 3D printed medicine. So it was really great indication for us that our dose plus can really produce amorphous or stable amorphous solid dispersion containing tablets, and that is truly commercially viable. Right, so I know I'm shorting of time, but I'll take 2 more minutes if that okay, and then hopefully I can stay a little bit longer if needed for taking up any questions. So this is another technology I just wanted to give you a little bit overview of. So this is SMART, which is sprayed multi-adsorb droplet repulsion technology. This is a technology that we invented last couple of years ago in 2020. So this technology is a new type of 3D printing technology for producing particles of highly sensitive molecules or macromolecules. So for an example, this shows how 3D printing technology can be used to produce particles in a cryogenic environment. In a left photo over here, we keep the temperature down to -80. If you look at the second photo over here, so here in a sterile vial, and here the temperature is kept at -170. So after producing this frozen particles, if you dry them up, that is how they appear. So they're really good application. This technology is really good for stabilizing vaccine formulation, and that can cut the cold chain storage significantly. So this recently we have filed a patent applications on that, and these are the comparative example or photographs showing the particle size. So here our target was to produce particle size below 5 microns for pulmonary delivery, and we're quite successful in controlling the particle size precisely, right? And we have recently proposed this to the FDA, CBER, for the continuous production of vaccine candidate. And we have recently been successful in getting a grant of about $1.5 million for continuous setup of this device and technique. And I think that will be truly revolutionary if we can optimize it successfully. It was recently highlighted in a newspaper. So now, okay, I have given various examples, right? Starting from the very basic personalized concept through the commercial exploitation or truly commercially viable technologies, right? So now 3D printing can actually help you to make it happen, like especially during the dire need, right? So we have used our 3D printing strategies for various applications during the pandemic outbreak, especially for producing PPE as well as modulated drug delivery systems and COVID test kit, right? So these are some news paper cuts taken from various media published over the couple of years, especially during the pandemic outbreak. And I would like to show you these 2 examples. One is how 3D printing can be really used to fight against COVID-19. Here we produced nasal swabs for ramping up our facility. we were able to cut down the production cost by 8 times, and we could easily ramp up the facility. So 3D printing can really be used during the dire need situation, like a pandemic outbreak that we have been gone through over the past couple of years. And through this example, what we concluded that by optimizing 3D printing concepts suitably, you could use less materials efficiently, but you can come up with a smarter solution to overcome some of this health crisis during the dire need when everything is completely shut down, right? The second example is about the personalized doses, like a pill. We came up with this concept called Model. This is multi-drug containing antiviral tablet, basically. We wanted to have a more personalized delivery of various antiviral drugs. And it was initially sponsored by, in a phase 1, basically, it was supported or successful for a grant through US Department of Health and Human Services. And in this model implemented digital manufacturing, we concluded that we use less materials for longer lasting effect in a smarter way. So these are 2 examples of showing the real time, real scenario based application or real, case studies where we used and implemented 3D printing technologies for combating a pandemic like COVID-19. And with that, I would like to summarize my talk in 1 sentence. And the sentence is that, okay, we believe that we agree that there has been a lot of research going on, and there's been done a lot of things been done, and I'm sure in the upcoming days more to do, but still there is still a long way to go in 3D printing to advanced drug delivery. And that is my 1 sentence summary. And this is the fun fact. I asked the same question to our daughter, because we have 1 daughter, and that was her expression when I asked her that question. So I don't know whether she agreed with me or not, but I leave that to you to judge. And finally, I would like to just acknowledge my group members, my family. Without them, I cannot do whatever we have been doing, basically my daughter and my wife, my group members in the US and in the UK, as well as my sponsors, supporters, collaborators, without whom we couldn't really achieve anything, or we couldn't really achieve whatever we were supposed to achieve or whatever we wanted to achieve. And most importantly, thank you very much for your concentration, your patience, and your support towards this webinar. I know I have taken a little bit longer than expected, but as I said, like, I'm more than happy to stay a bit longer if needed, and I'm happy to take any questions if you have. Thank you.
Thank you so much, Dr. Maniruzzaman, for the wonderful presentation. And I was really impressed with not just the quality of the research, but also how you're managing to find solutions to the challenges of the 3D printing. Like, for example, the SLS technology, I think it really has a huge potential, and it will definitely improve the commercialization challenge regarding the 3D printing material. So thank you so much for this one.
You're more than welcome, and I totally agree with you.
And I think we have time for some of the questions from the. So I would kindly invite the attendees, if any one of you would have a question to Dr. Maniruzzaman, just raise your hand and we'll take the questions. We can start with Mohammed Salah. Please go ahead.
Hi, Dr. Maniruzzaman. I want to first of all, I appreciate this talk. It's a great talk and a great lecture. I have some questions just to understand more. I'm Dr. Mohammed Salah, lecturer of pharmaceutics and industrial pharmacy at Helwan University. My first question: what about the stability of these 3D printing forms? What are the conditions you test for stability?
Okay. All right, good. So that's really good questions in terms of the stability, right? So now for the 3D printed doses forms, like if you see the case studies that I presented, at least 2 different case studies, normally for the 3D printed product, the stability conditions or the experimental setup will be same as the traditional doses forms, right? Even though I'm sure that you will agree that there is no set protocols from the FDA yet, because 3D printing is still a new domain for the regulators worldwide, right? So we just wanted to compare an apple for an apple. So when we compare with the traditionally formulated doses form, we just keep the same condition, right? But there is no specific guidelines for the stability study for 3D printed medicines. The reason is that because it is new. And the second thing is the regulators worldwide, like FDA, EMA, or even MHRA in the UK, they're still developing guidelines around that, right? So yeah, as of now, we have used the conventional approaches available for conventional dosage forms.
Yeah, so if we talk about this point, can we say we can use the guidelines of the non-stability conditions, but we have some concern about, this 3D printing may expose the drug and the polymers for more aggressive, maybe slightly temperature, maybe the heat from the laser. So do you think we can adhere to the guidelines of the normal conditions of the stability, like temperature and humidity, but with this slight concern about these other conditions during manufacture? It will work.
That is, again, as I said, like it is really difficult to answer, right? As I said, like there's no set guidelines, but for whenever, say for example, whenever we are promoting or proposing a new technology, let's say if you consider the commercially available jeep dose based sputum medicine, right? If you I don't know whether you had a chance to look at it, or if you just take it, you will see they're very porous, very fragile, right? So now if you just keep it on your hand or on the palm, you will see they will start degrading, kind of like they will start breaking, because they're really fragile. They're highly porous tablets, right? So now if you wanted to do the normal quality control check with that tablet, as you would do for the conventionally manufactured tablets, you will see they will not even withstand any friability testing, because they will break, right? But yet they are approved by the FDA because they are not considered as the traditional dosage form. So I think in terms of the stability and quality control of the additively manufactured tablets of dosage forms, I think regulators are still working. In fact, we know that we have collaborators in the FDA, in the CPQ, OPQ, like CEDAR branch in the FDA, and we know they're continuously working towards developing a framework that could be really widely applied for additively manufactured dosage forms. So I'm sure that in the next few years or so, there will be some strict and concrete guidelines around the quality control, including the stability and other properties.
Yeah. Okay. Thank you. So my next question, for the multi-loaded tablet, what do you think about the interactions that may occur between the drugs after release? We may have the same interactions that will occur from the multi-dose that occurred before before the DCD printing. Okay. Yeah. Or maybe we have more interactions as the 3 drugs or more than 1 drug is incorporated in the same device. It may occur, or you study something like that, yeah?
Yeah, because there is a high chance, because we actually I was part of reviewing one interesting study where multiple drugs were used in one polypill system. So polypill is basically where you combine multiple drugs into one, right? There's a high chance that drug like unexpected or kind of like unwanted drug interactions or reactions may happen, especially if you talk about that photopolymerization based printing. But there are ways, like, so one thing that we are supposed to do, and what we do basically when we apply multiple drugs into one device, we try to make sure that there is no drug interactions, because otherwise that will alter the pharmacokinetic profiles after the delivery, right? So this is one of the things that we have to look after. Now there are ways, of course, to overcome those challenges. One could be by separating separating each layer, or like separating layers of 2 different drugs by applying a pseudo or a blank layer so that they do not get in touch, right? So in this way, at least you'll be able to eliminate some potential interactions during the printing process.
Yeah.
Now, once you take
multi-layer batch, yeah.
So once you take the tablet orally, once it goes into stomach, there may still be some interaction, but again, that will not be different than taking 2 tablets separately, because if you take 2 tablets separately, they will go into the stomach anyway, right? So at least we can justify our rationale by creating that pseudo or a blank layer, which can eliminate some of the potential interactions.
Yeah. Yeah. Okay. Thank you so much.
Thank you.
What about my third question about the some changes? And you said that we have better distribution rate in this new device and new 3D printing. So it shows the difference in bioavailability. This difference in distribution rate affects the bioavailability of these drugs, increases or decreases or something like that. Did you study something like that?
No, we haven't. But if I got your answer right, because your voice was breaking in between, so if I got it correct, you asked me whether there was any difference between the bioavailabilities of two different drugs, correct?
Yeah. No. Most of these changes show changes in distribution rate. Yeah, you get better distribution rate. So did you get a different bioavailability due to the difference in this distribution rate?
Okay, no, we haven't really done any animal studies yet, so we have not done on at least those examples that we showed, but there are some studies where animal studies have been conducted, especially in different models, rodent models, and where there was clear evidence of the bioavailability enhancement. Yeah.
Okay, excuse me for my last question. For the 5-fluorouracil loaded magnetically triggered sponge, this is like it applied in like cell line, yes? Or what do you think about how we can trigger it inside the human body when we go to the clinical trial?
Yeah,
how we can make a magnetic triggering for this inside the human?
Yeah, that's really good question. Yeah, so I think the idea was basically to have a magnetic field outside the body. Like say this can be implanted. So these are like implantable small tiny device. So which is like an implantable drug delivery system like other delivery systems. The only difference is since they are magnetically sensitive, so they are loaded with magnetic nanoparticles. So if you insert underneath your skin or near the site of delivery or site of action, so if you bring a strong enough magnetic field close to the implant, that will actually is a kind of like contract the magnetic sponge inside the device, and as a result of that, some drug will be released. So the moment you want to release the drug, you have to keep the magnetic field close to the body, and that will release the drug for a certain amount of time. And then if you take the magnetic field away from the body, then it will close the gate. We are also currently working on developing a new, it's a kind of like oscillating magnetic field where you can actually manually increase or kind of like incremental opening of the or incremental availability of the magnetic field to the body, close to the body. So you can actually control the magnetic field switch basically. So this way, these were our hypothesis of coming up with this 3D printed magnetically triggerable drug delivery systems. But there may be other ways of delivering it in the practical scenario, but we don't know what that would be yet. But our initial idea was to have the magnetic field.
Thank you so much for your time, for your question, and secondly, I appreciate the lecture, appreciate your time. It was amazing, like presentation, and good luck for your work. Yeah, thank you so much. Thank you.
Thank you very much for your question. Thank you.
Thank you, Dr. Muhammad. The next question is by Dr. Nahla Ahmed. Please go ahead, Dr. Nahla.
Okay, hello everybody. Hello, dear professor. Thank you so much for the amazing lecture. Thank you, Dr. Nasr, for this very beneficial webinar. Actually, I have small questions I'd like first to ask about if the 3D printed tablet avoids the first pass metabolism, gastric upset, and food interaction, mainly avoids the disadvantage of the traditional tablet, or it can happen.
Yeah, so it really depends on what sort of delivery route you're choosing. So now if you look for oral delivery, then of course you cannot really avoid the fast metabolism, right? So there is a if it is a traditional standard oral delivery, then yeah, fast metabolism is the barrier that you have to go through. But there are other ways to overcome that. So for example, with the 3D printing technology, you can have a buccal delivery based dosage form, or even or even say, for example, the container system example that I gave, there we can have a kind of more anchoring system at its rate. So that whenever the container system actually goes to intestine, we can keep it there for longer periods so that most of the drug will get absorbed through the epithelium layer of intestine. Right. So there are ways. So now there is another project that is currently going on where we are trying to keep the dosage form afloat in our stomach or in gastric juice for a couple of weeks. So in this case, the idea is basically that there's a high chance that drug will get absorbed as they pass through the intestine, right? So that, hopefully sufficient amount of drug or just enough to be showing enough to show the therapeutic effect after getting absorbed through the epithelium intestine. So yeah, in a short form, to answer your question, it really depends on what you want to achieve at the end. So if you wanted to go for alternative route of administrations where fast metabolism can be avoided, it is possible to do with the 3D print. You have ample opportunities. You can basically go with any kind of geometry modification. You can play with the formulation, process parameters, and yeah, it really depends on you. So there is the sky is the limit, we say, or actually we say like sky is the starting point for us for innovation. So you can do a lot of good things with 3D printing. Yeah.
Okay, great. That's why my next question is the powder produced from the 3D printed machine can be used in the topical route, transdermal, or just it will be restricted for buccal, for floating tablets and so on?
Yeah, as I said, like with the 3D printing, you can literally do whatever you want to do, right? So for example, we have projects where we use 3D printing for gel. We are using 3D printing for transdermal drug delivery. We are using 3D printing for topical delivery of medicines. We are using 3D printing for medical devices. We are using 3D printing for contact lens, like for ocular delivery. So the opportunities are huge. It really depends on what you want to achieve at the end. So there is another kind of 3D printing techniques that is known as semi-solid extrusion printing, like a semi-solid extrusion. So where you can actually use hydrogel as a starting material or a printing ink, and then you can produce gel or scaffold or any kind of soft device based on your application area. So yeah, in summary, the answer is yes. You can use this technology for topical delivery now.
Great. Just to facilitate for me the imagination of 3D printed machine, is it look like electrospinner? There will be like a solution in a nozzle and something like spraying. In the video you showed for us, it was I imagine that it like a solution sprayed in a specific area. So is this imagination is right, or I'm away from the?
Your imagination is partially right. Yes. So that's why called SMART. It stands for sprayed multi-absorbed droplet reprocessing technology. So the underlying concept in a simplified form is correct, and your imagination is absolutely to the point. But there are other things that we have to do at the background. So we couldn't really show you the whole video, but yeah. So but in a simplified form, whatever you have projected is correct.
Okay, thank you so much. Thank you, Dr. Maha. Thank you, Professor.
No problem. Thank you.
Thank you, Dr. Nella, for your question. I think we have a time for one last question. Dr. Hamdi.
Okay. Thank you. Thank you for the organizers and the speaker, and very interesting talk, and very good, very nice topic. My question to Dr. Maniruzzaman. It is not a typical question, but I would like to briefly comment on different types of excipients or filaments that can be used with 3D printing tablets. Are they available on the market from Sigma, for example, to order, or you have to prepare it in-house? Okay. That's it. Yeah, that's my question.
That's really good question. So it's basically in the market, there are some vendors, like as you mentioned, like Sigma, for example. Sigma, they don't sell filaments as of now, but there may be some. But there are other commercially there are some other vendors who sell various pre-formulated filament for FDM-based 3D printing, right? Now, one major problem there is that those are not necessarily suitable for pharmaceutical applications. There are some PLA grade polymeric filaments or PVA grade polymeric filaments, which are pharmaceutically acceptable. You can use it for pharmaceutical application. But if you needed to use those filament for your specific applications, then we always recommend that you produce your own filaments, right? So that you can make your own composition, drug polymer composition, and then you produce this feedstock material for the tunable or modified applications that you are looking for. But now, as of now, we know some vendors, they sell PLA grade or even medical grade filaments, but not necessarily they may be suitable for your applications you're looking for. So in this case, we normally recommend to prepare your own composition.
Okay, thank you.
Thank you, Dr. Hamdi, for your question. So I think we've now came to the end of our session today. No more questions. Thank you, our distinguished speaker, Dr. Maniruzzaman, for the wonderful and interesting talk. And we are looking forward to welcoming you in person in Egypt and looking forward to more collaboration with you in the future. And again, I'd like to extend my thanks to Professor Dr. Sameh Surur, the Dean of the Faculty of Pharmacy, Helwan University, for the kind invitation. I'm always honored by our friendship and our continued collaboration. And thank you so much.
All right, thank you very much. Oh, sorry, you go on, please.
No, just go on. I will just finish.
I just wanted to take this opportunity. Thank you all for organizing this well-presented webinar. I really enjoy it, communicating and coordinating with you, and especially those who asked me some interesting questions. Thank you very much for your patience. And I'm sure, as Professor Nasr mentioned, that I'm looking forward to further collaborations between these two countries. And as you already know, that we are hosting 5 scholars from your country, from at least